Good evening, people. Can you hear me? Can you hear me clearly?
Can you hear me? You can hear me, right? Am I audible? I'll.
Right, people. I have given you permission to chat with everyone. Hope you will chat with me using that. People, can you hear me clearly? Can you hear me clearly now? Yes. Yeah. Okay. Yes. Right. Now, today's discussion, paper 8, people, I gave you this paper 2 weeks ago. So you had enough time, I think, to answer the paper. And I'm sure you have answered the paper, right? You are such a nice set of students, I know. So you have answered the paper. So we'll go through the marking scheme, people. We'll go through the marking scheme. But we'll start first with the essay part, essay questions. I think that's better, neither. Normally we start with the questions, MCQs. Today we'll start with the essays, people. So essay question, we'll go with the first essay. Question is regarding describe the behavior of chromosomes. Do you have the marking scheme, all of you? I distributed the marking scheme last time. Do you have the marking scheme with you? Anyone wants the scheme? Telegram. All of you have? Yes, sir. Yes, sir. And anyway, I'll upload the scheme and the paper. Right, people, we have to discuss the essay part first. Essays. So check this essay. The essay question is about describe the chromosome structure. Describe the chromosome behavior of chromosomes. Describe behavior of chromosomes during mitosis. Describe the behavior of chromosomes during mitosis. Now, chromosome behavior during mitosis. You know, people, mitosis is the nuclear division. So, can you tell me the phases of the mitosis? Prophase, metaphase, anaphase, telophase. Neither. Those are the phases. Dual audio. Okay, I know how to fix it. Hari. Thank you. Is it okay now? Is it okay now? Is it okay now? Please send a message one more time. Venuk. Now okay, ne? Hari, fine. I couldn't finish setting up this studio. There are certain things that I need to permanently fix rather than adjusting it every week. Yeah, I couldn't finish it. Right, people. The behavior of chromosome. Now, if you take prophase, we can see that the chromatids, chromosomes become visible, cystochromatids become visible. We can see that they are attached. Now, chromosomes, cystochromatids attachment does not take place during the prophase, but attached ones can become visible, people, during the prophase. Then what happens, people? Then we can see that during the prometaphase, kinetochore come and bind. Spindle fibers are going to attach. And chromosomes are going to move like a swing forward and backward. And then they begin to align to the equator. So we know in the metaphase, chromosomes are going to come and align at the metaphase. Right? The spindle attachment is going to complete. Chromosomes aligning to the metaphase is going to complete. All chromosomes are going to be like attached to the kinetochore fibers and all are aligned to the metaphase plate. Then during the anaphase, we can see that the cohesin is going to break, sister chromatin are going to separate, and they're going to migrate to the opposite poles. That happens during the anaphase. And finally, we can see that at the telophase, people, the nuclear envelope reforms around the chromosomes, forming what do you call the nuclear envelope plus chromosomes transform back again into chromatin. So these are the points that you need to write here. These are the points that you need to write. Shall we go through those points, people? Chromosome fibers condense by shortening and thickening and transform into chromosomes. So chromatin become chromosome. Chromosomes appear with two sister chromatids attached at the centromere. Chromosome arms attached by special protein cohesin. Chromosomes get even more condensed in prometaphase. Special protein cohesin attached. Sister chromatids of each chromosome at their centromere, some microtubules that attach the kinetochore of the chromosome. Move chromosome back and forth. Now in the metaphase, the chromosomes have arrived the place called the metaphase plate. This is located at the equal distance from the pole. At the end of this fold, each chromosome is of this cell get attached to the kinetochore microtubules at the centromere and align at the metaphase plate. Anaphase sister chromatin are separated at the centromere. By the end of the anaphase, equal and complete set of chromosomes found at each pole. So we have 22 points in this answer. So we'll go to the other page. Let's go to other page. Then at the telophase, nuclear envelope reforms around each, nucleoli reappears, chromosomes unwind and become chromatid. So those are the points I have considered. Please understand, people, still you are not answering full papers. Then make a full papers kela jatiyan neme hari. Nikan questions kali tiyenne hari. We are gradually training you to people write answers. Now you check with the 2025 paper A-level students. We are discussing now the 26th paper I have given them today. And now 26th onwards, people, full papers. So from 26, another 10 or 14 papers will be discussing, people, depending on the time availability and their speed, right? From the remaining time, we expect to go for 40 papers, right? That's our aim. So now only they go with the full papers, right? So you are going to get pieces of papers, right? But that's good enough. My aim is to people rotate the sections again and again for you to answer similar questions so that you can study repeatedly until people these information, right, properly registers in your mind, right? So my aim is to take you through the same theory part with different questions again and again multiple times, so that gives you the chance to study. All right? Okay. Next one, people, write short notes on the following. So don't worry that the questions are like partial, so 22 points නෙවෙයි 38 points දෙනවා නේද විභාගයේදී. එතකොට ඉතිරි points වල මොකද උනේ? එච්චර හිතන්න එපා. හරිද දැම්මම. කොහොමද සර් point breakdown එක යන්නේ? We'll check them later, okay? You can now take people 150, so that is like 7.5 marks for each point, right? 22 20 කියලා ගන්නකො. Right? So we can go properly giving you marks, people, later, not now. Write short notes on the following. Significant features of seed plants, right? Seed plants. Now, what are seed plants? Plants that produce seeds are the seed plants. You know, seed is a structure with an embryo and endosperm which is acting as the propagating structure, right? So that helps the species to disperse. So what are the special features that you can see in seed plants? First one is they are producing seeds, right? They produce seeds. And then people in the seed plants, the gametophyte have reduced. So that the gametophyte is reduced and then hidden inside the sporophytic tissues. Gametophyte reduction. What else we said people heterospory condition. What is heterospory? People, production of two types of spores. The two types of spore production is not a feature begin from the seed plants. What is the first phylum to show heterospory condition, people? Which phylum to show heterospory condition first? It is Lycophyta, right? Lycophyta. Now, Bryophyta is homosporous. Pterophyta is homosporous. We can see that started from Lycophyta. Lycophyta also spike mosses and club mosses. Club mosses are homosporous, spike mosses are heterosporous. They start from Selaginella, right? But you can see that this situation continue when come to the seed plants. All seed plants are heterosporous. All seed plants are heterosporous. Then they produce ovules in eggs. Egg is produced inside the ovule, right? So it's today I'm not going to take any attempt to describe what ovule is and all these features, right? You have learned it. So it's your responsibility to go through the notes and then check these things once again. We have learned these things with life cycles, people. Life cycle print kala poster karla dila ne. I have to give you 2 more life cycles. I'll give them as soon as possible, right? So production of ovules and expression inside the ovules, right? So these eggs are produced by the female gametophyte, which is present inside the ovule. So what you learn there, female the ovule inside the nucellus present. Nucellus has the spore mother cell. Spore mother cell undergo meiosis and then produce 4 spores. 3 are going to die, 1 remain. Remaining megaspore is going to grow into the female gametophyte with archegonia. Seed plants have archegonia. It was a production of pollen and sperm. Pollen is the spore, microspore, produced by the microsporangium. This pollen inside, you can see people the male gametophyte present. Male gametophyte present, pollen inside the pollen. Okay. So these are the points that I have allocate marks, my dear students. Right. These are the points we have considered for marking. Right. Look at these production of seed. Seed consists of an embryo and endosperm. Sorry. Because when you write only neither, these things are going to register. You write multiple times because you have enough of time neither. Endosperm suddenly supply food to the embryo. These endosperms surround by a protective coat, the seed coat. Reduction of gametophyte. Tiny gametophytes develop from spores and they are retained within the sporangia of sporophyte. These gametophyte protects these arrangement protects the gametophyte from environmental stressors. This relationship also enables the dependent gametophyte to obtain nutrients from the sporophyte. Heterospory. Seed plants are heterosporous. Seed plants are heterosporous. Each megasporangium has single functional megaspore. Each megasporangium consists a large number of microspores. Production of ovules. A layer of sporophytic tissues called integument covers the megasporangium. The entire structure containing megasporangium, megaspore, and integument is the ovule. You can remember the Seeker's life cycle, ne? Right? If you just memorize people this Seeker's life cycle, you can easily understand these features. Inside each ovule, female gametophyte develops from the megaspore, production of pollen and sperm. Megaspores develop into pollen grain that consist of male gametophyte. The transfer of pollen grain towards the ovule is called pollination. When pollen germinate, it gives rise to pollen tube that discharge sperm. Inside the pollen grain, sperm producing male gametophyte present. Right, then people, I want you to write a short note regarding sclerenchyma. You know, sclerenchyma is a cell type present in ground tissue. Ground tissue has parenchyma, collenchyma, sclerenchyma, ne? Right? And then what else you know? Sclerenchyma cells are lignified, dead cells, lumen present, no cytoplasm, cell walls have pits, no intercellular spaces. They provide support. Dead cells, the two types of sclerenchyma cells are there. One of the cells are sclerates present, fibers present. Secondary cell wall. Secondary cell walls produce after cell elongation. They have secondary cell walls. They are dead cells at maturity. Two cell types, sclerates and fibers. Shorter and wider cells than fibers. The sclerates irregular in shape. Very thick lignified secondary cell walls. They are found in spaces where the growth has stopped. Fibers are usually grouped in strands. They are long, slender, tapering. Used commercially to obtain fibers, coconut husk fiber and all. Right? Oh, that's all, ne? Okay, that's all for the essay paper. Now, people, what do you think better to question in the next paper? One of the you have to tell me, people, the sections you fail to study reason. Then it gives you an opportunity to go through that. Ne? What sections? 3 sections we have to question, people. What 3 sections you think is suitable to question next part? Tell me 3 sections to question. Now we did cell cycle. Make a plant plant primary growth name. It was say then we question you people respiration. Ne? Cell respiration is lamai. Cell respiration ne? Cell cycle. Cell cycle. Plant diversity ne? Plant diversity and yeah. Secondary growth handle. Secondary growth handle. Mko dikhiyan ne. Secondary growth common athi ilanga part secondary growth niya. Secondary growth. Now, secondary growth from unit 4 ni. Unit 3 ke manad danna. Unit 3, send it to the main chat. Fatima says secondary growth. Unit 3 ke manad danna ni. Unit 3. Come on, tell. Unit 3. Come on, tell me. Phylum Nidaria. Animal phylum. Now, abhi unit 3 ke main dam. Unit 3 ke lamai invertebrates ke dam Invertebrates. Invertebrates. Ekane Nidaria, mollusca, nematoda, platyhelminthes, Aeticockum. Hari invertebrates, not vertebrates. Invertebrates. ඊට පස්සේ unit 3 එකෙන් මොනවද දාන්න? Unit 3 cell organelles ලෝ. Utica says cell organelles. Cell organelles වලින් අහමු දෙනවා. හරි එහෙනම් cell organelles. එතකොට paper 9 you're going to get this Saturday. හරිද? Paper 9 එකට ඔය ටික එන්නේ. හරි paper 9 then these 3 sections Okay, we'll go like that slowly, slowly, steadily. We'll go on doing questions. No need to hurry, but then no need to rest also. We'll do both slow and steady. So that's going to take you to the goal. Right? Now, these days, people, 2025 batch, some students are there who had answer the papers continuously, people, they are doing a good job now, right? They know each and everything, right? All the tiny things in the syllabus, they have gone number of times, so they answer very accurately, right? So that's good, ne? Okay, so you can come to that the same level, right? It's a slow process. You make mistakes and you have to go slowly. Don't worry about it, right, people? But we have to get the other people to this job. අද අපි මේ ළමයි ටික වෙනම රෙකෝඩ් කරගෙන දෙමු සෙනසුරාදාට ආපු නැති අයට නේද? මොකද කියන්නේ? දෙමුද ගුටි එන්නේ නැති අයට. ඒකට මොකද කියන්නේ? දෙමුද එපාද? පවුද? මොකද කියන්නේ? දෙමුනේ. We'll punish them නේ on Saturday. Right? ඉන්න බැරි එකට දෙමු හොඳට රිදෙන්න. Right. So we had to go with the structured essay paper and the MCQs. We'll go with the structured essay paper first, people. Structured essay paper, first question. Structured essay paper, first question. So cell cycle, it's not from unit 3, but from the second unit. Yeah, second. Yeah. Really? Right, we'll move to the paper. We'll move to the paper. We'll start with the structured, first structured question, people. We'll start with the first structured question. Draw a chart to show relative proportions of phases of the cell cycle. Proportion over the phases of the cell cycle. So you draw this chart. Phases of the cell cycle. This one. So it will show G1, S, G2, and M and cytokinesis. Right? Those are the phases. Are you going to give 9th paper this Saturday? Yo, එක තමයි plan එක. 9th paper එක මේ සති දෙන්න. Right? එතකොට next Wednesday අපිට වැඩක් නැහැ. Week after we can take the discussion නේ. Marking scheme දුන්නම. Right? I'll give you 1 week to submit. Right? So I think you mark this. No issue, ne? So how many marks, people? 8 marks for that. Then what is the difference between cell division and nuclear division? Cell division the cell division is the nuclear division plus cytokinesis is a cell division. Formation of 2 daughter cells from the division of parental cell is cell division. Formation of 2 nuclei is called as nuclear division. State 3 phases of the interphase G1, S and G2. State main events occur during the S phase. State main events occur during S phase. S phase DNA replication occur. Synthesis of histone protein take place. And then DNA and histone protein, they form chromatin because you know the DNA present as chromatin during this state, right? So you can give people any 3 marks, all 3 marks. Ne? Next one, people, state 3 checkpoints G1, G2 and M. 3 marks for that. Which of the following statement are correct regarding interphase? Interphase nuclear envelope absent during the G2 phase. Nuclear envelope absent during the G2 phase. What do you say about that? Nuclear envelope absent during the G2 phase. No, G2 phase has the nucleus. You know, nuclear envelope disappears during the prometa phase. So until that, nuclear envelope is present. So this is wrong. And then centrioles duplicate during the interphase. Centrosomes are duplicating during the interphase. That is correct. That specifically happens during the G2, ne? Microtubules are attached to kinetochore of the chromosomes. No, that doesn't happen during the interphase. The microtubule attachment occur during the prometapase. So that is a wrong statement. Nerve cells and the muscle cells at G0 stage, that is correct. Chromosome appears as two sister communities attached at the centromere. Chromosomes appear as two sister communities attached at the centromere during the interphase. Chromosomes appear as two sister communities. Wrong, neither, because they are attached at the centromere, no doubt about it. But thing is, people, they are not visible as sister communities. The sister communities become visible only during the prophase. So that is wrong, not during the interphase. What happens to a cell if it does not receive a go-ahead signal? You know, people, when it does not receive a go-ahead signal, the cell enters into the G0 phase, where the cell undergoes specialization and then carry out people normal function inside the tissue, right? What is a cell type has G0 phase, nerve cells and the muscle cells. State different functions of cohesin protein and kinetochore proteins. State special functions of cohesin protein and kinetochore proteins. Cohesin proteins attach the chromosome arms of the sister chromatids together. They are the cohesin. So their job is to hold the sister chromatids together. Then what is kinetochore, people? Kinetochore is a protein disc that helps in attachment of the spindle fibers. All right? Spindle fiber attachment. එතන ලකුණු දෙකක් මන් දීලා තියෙනවා. Arrange following events occur in prophase to correct order. Arrange following events in the prophase. Centrosome move towards the opposite pole. Chromosome fibers get condensed. Spindle fibers form. Chromosome become visible under microscope. Right, we'll take the first one, people. Chromatin fibers get condensed and then chromosomes become visible as a result. Ne? ඊට පස්සේ spindle fibers are going to form. Finally, I think they have given the centrosomes move towards opposite pole එක අන්තිමට දීලා තියෙන resource එකේ. Right? So you had to know them, people. Sometimes you learn these things, but you don't know, you're not sure about the order. So order is important too. So how do you memorize this order and everything, people? Just repeatedly check with the questions. You have to check that, right? It doesn't matter that you go wrong, but then what important is for you to check people again and again and again. So you will learn more and more, people, from the same content. You're going to learn more every time you learn, right? Identify the following two diagrams. First diagram is metaphase of mitosis, because you can see that the homologous chromosomes, now this red short and the black short, they are homologous, but they don't pair. Therefore, this is mitosis. How many chromosome pairs here? Two chromosome pairs, mitosis. Mitosis of two chromosome pair. This cell is, people, mitosis of two chromosome pair. This is given for mitosis of two chromosome pair. Two pair of chromosomes. Now this one is metaphase of three chromosome pairs. A cell with three chromosome pairs. Make three chromosome pairs. This is a cell with three chromosome pairs. This is a cell with maker, right? This is a cell with three chromosome pairs. This is a cell with two chromosome pair. Cell with two chromosome pair. Okay. So Right, people? We'll move to next question. State difference between microtubule attachment and the chromosome. State difference between microtubule attachment and chromosome arrangement between these two stages. Metaphase of mitosis, metaphase. Difference between microtubule attachment and chromosome attachment between these two stages. Again, metaphase of mitosis and metaphase of meiosis 1 lu. Right. Now, metaphase of mitosis, people, you can see that the chromosomes are arranged in the metaphase plate, and then metaphase 1 of meiosis 1, pairs of chromosomes are coming and aligning at the metaphase. That makes a difference. Ne? Now, you know, in mitosis, no homologous chromosome pairing. They pair during meiosis, and they arrange in a way that the centromeres are located at the metaphase plate. Here, people, they are arranged in a way that one chromosome of each pair face each pole. Ilangata, chromatids of each chromosome get attached to kinetochore microtubules. Here in metaphase 1 of meiosis 1, both chromatids of a homologous chromosome attached to the same pole. In mitosis, you can see chromatids are attaching, sister chromatids are attaching to opposite poles. But in meiosis, both sister chromatids of a chromosome attached to the same pole. So those are differences. What is the phase of attachment of the spindle fibers to the sister chromatids during meiosis? What is the phase of attachment of spindle fibers? Meiosis 2. Meiosis 2 ke there is no prometaphase. Ne? So that happens during the prophase 2. What is meant by metastasis? Metastasis means, people, the cancer cells, spread of cancer cells from the location distant from their origin site. Ne? They origin at one site, and from there the cancer cells are going to spread to the other tissues. We call as metastasis. Right, next one, people, second question. Second question, what are meristems? Now, you know, people, meristems are undifferentiated tissues consisting of cells that constantly divide under suitable conditions and produce new cells. State 3 characteristics of meristematic cells. Right? They are living cells, isodiametric, structural and the functionally undifferentiated. They have central nucleus. They have dense cytoplasm. They have the ability to divide. Draw the root tip and label. Draw the root tip and label. These parts must be there, people. What are those parts? Oh, epidermis, cortex. Actually, draw the root tip Q root tip and the apex. Apex nang me apical meristem mega. Root tip ki all these three zones together. Zone of cell division, zone of elongation, zone of differentiation, everything together we can say the root tip. So zone of cell division has the root cap and the apex. Zone of elongation, cells are elongating. When come to differentiation, root hairs present. In addition to that, vascular cylinder, cortex and the epidermis can be observed because the other tissues are now differentiating here in this region. Hari? Moving to next page. Next set of questions. What are leaf primordia? Finger-like projections alongside of the apical meristem. Part B, identify A and B diagram. What is A? A is, can you identify A, people? This is collenchyma. B is parenchyma. Now, how do you identify parenchyma? You can see large intercellular spaces are there. What are these, people? Intercellular spaces. Intercellular spaces present. They are large in parenchyma. They are not present or very small when come to collenchyma. So you can see clearly, people, this cell wall thickness difference also. Here you can see that the cell walls are thick at the corner. The cell walls are thick at corners like that. Parenchyma, you don't see that. Uniformly thickened cell walls present. Indicate how B structure is different from A structure. What is B structure? B is parenchyma. How parenchyma differ from collenchyma? Parenchyma have relatively thin flexible primary cell walls, have large central vacuoles and evenly thickened cell walls, but you don't see them in collenchyma. Collenchyma have relatively thick walls and then they have vacuoles anyway, but then they have unevenly thickened walls, especially the corner walls are thickened. How functionally C and D different? C is, what is that people? C is vessel element, neither. Xylem vessel element. What is D people? D is a trachid. D is a trachid. How functionally C and D differ from each other? In C, water flow freely through the perforated plates. Okay, here we can see that these are perforated plates, ne? Perforated. And here we can see that the pits available in the cross walls, ne? Pits present. Functionally, how they are functionally different? State the cells properties of the C tube element. State cell properties of C tube element. Lac nucleus, ribosomes, a distinct vacuole and cytoskeletal element. Lac nucleus, C tube element. No ribosomes, no cytoskeletal elements. Cytoplasm reduced to thin peripheral layer, consists of sieve plate. State function of the companion cell. We learned that the companion cells are helping in functioning of the adjacent C tube element, especially people from loading and unloading, they play a role. Ne? So they say that nucleus and the ribosome of these cells serve to the adjacent C tube element, help in from loading and unloading, right? Those are the functions. Then part C, state vascular plant phyla, vascular plant phyla. Vascular plant phyla මොනවද ළමයි? Lycophyta, Pterophyta, Psychotophyta, Coniferophyta, Gnatophyta, Anthophyta. State a difference between club moss and spike moss. Spike mosses are heterosporous and club mosses are homosporous. What are microsporophylls and megasporophylls? Microsporophylls, single veined, small size leaves. Megasporophylls are large, flattened leaves with branch veins. Then complete the following dichotomous key. People, you know how to prepare a dichotomous key, right? Then dichotomous key, I think we discussed that some time back. Hope you have not forgotten. Every statement has two. Sorry, every step has two statements, present, absent wage statements. So let's see how to complete this dichotomous key. If you take people, cucurbit කියන්නේ flowering plant එකක් නේ, වට්ටක්ක, pumpkin. Anthocerus, that is bryophyte. Coconut, marcantia. Coconut is monocot, marcantia. Seedler vascular plant, sorry, bryophyte. Lycopodium, seedler vascular plants. Netum, seed plant. Cecus, seed plant. Complete the following dichotomous key. Complete the following dichotomous key. I want to share layout. I want to go to whiteboard, layout, uh, side by side. I want to use whiteboard, whiteboard. Paint එක වගේ යන්නේ නේද? Can you see this paint? Can you see this paper? Can you see the paint or not? You can see ne? Sorry, මෙන් බලමු මෙතන දැන්. Okay. දැන් මෙතන තියෙනවා cooker bit. Cooker bit. Sure. ලස්සනයි නේ. Anthocerus. ඊට පස්සේ අපිට තියෙනවා coconut. Then we have people Markantia. Markantia. ඊට පස්සේ we have people Lycopodium. Lycopodium. Then we have people Netum and then Cycus. Netum and then finally we have Cycus. So what do you do now? All these plants we going to divide into non-vascular, no vascular tissues and vascular tissues present. දෙකට කඩම් බලන්න. How to take that? No vascular tissue. කවුද ඉන්නේ no vascular tissue? No vascular tissue you have people Anthocerus. Antho. ඊට පස්සේ you have Markantia. Markantia නේ. And then you have people, that's all නේ. ඊළඟට the other side vascular tissues present නේ. Vascular plants. What are vascular plants? We can say cucurbits. And then coconut. Lycopodium. Netum. And Caecus. So this is my first dividing step. This is 1A. This is 1B. So 1A goes to number 2 ne. 1A people has to go into number 2. So number 2 we divide further. How do we divide number 2? Liverwort and hornwort. Liverwort and hornwort. Who's liverwort? Markantia. Who is hornwort people? Hornwort is Anthocerus. Anthocerus ne. So then that branch is over. Then this one 1B goes to 3 ne. Step number 3. So you write people 1B as number 3. Then we divide these people based on these two characters. One of the seed, no seed, seed present. No seed. And what's the next one? Seed present. Seed present. No seed, seed present. No seed, seed present. No seed, I am taking people Lycopodium. Then all the others are having seeds. Cucurbit, coconut, netum, and cecus. Right? Only lycopodium. So what you can write, people? No seeds, you can write lycopodium. When you have one member, you can write the name. So this is, you can write the name now. Seed present, that is number 3B. This is 3B, this is 3A. This is 2A, 2B. So 3B now divide. 3B is number 4 now. Number 4th step now. 4A open seed. Open seed. 4A. How the open seed lamai? Open seed, you can take netum and cecus. Netum and cecus. Then 4B, what do you call, people? Fruits present. Fruits present. Fruit present, that is number, key the number. Open seeds key the number. Open seed plants number 5. So we can divide further. We can divide this further into 2. We can divide this further into 2. How do you divide that? Open seed, we can take, people, number 5. And gymnosperm with xylem vessels. Xylem vessels present. Now who has xylem vessels? Netum. Then palm-like leaves. Palm-like leaves. Kauda. Cecus. Then number 4B. Fruits present number 6 world. It goes to number 6. 6, then we have 2 branches. 6A mukadlamai perianth not differentiated. Perianth Who's having perianth? Coconut. And then people, calyx corolla present. Calyx corolla present in cucurbits. Present in cucurbits. Right? So when you are writing the, what do you call when you're writing the dichotomous key, now this is not the dichotomous key. This is just a plan to make the dichotomous key. So you given the dichotomous key, people, now you have to fill that. So how did you fill that? 1A, 1A, what did you write? 1A, you wrote number 2. 1B, what did you write? Number 3. 2A, what did you write? 2A, Markantia. Let's check that given answer now. So look at this. Right? 2A, liverwort, Markantia. Anthocerus. Okay, this part. 12 marks, 12 marks. You can't draw that, what you call, tree diagram. This is what you have to fill. Right. Then people, we are moving now for multiple choice questions. MCQs. Let's move to multiple choice questions. MCQs. Right, first question. Select the correct statement. Select the correct statement about eukaryotic nuclear division. Eukaryotic nuclear division. Nuclear envelope is fragmented and centrosomes are located. Centrosomes are located in the metaphase plate during prometaphase. Nuclear envelope is fragmented and centromeres are located in the metaphase plate. When the band line wrong ne? Kinetochrome microtubules move chromosomes back and forth during anaphase. Wrong. That happen in the prometaphase. Both chromatids of chromosomes are attached to kinetochrome microtubules from one pole in prophase 1. Both chromatids of one chromosome are attached to kinetochrome microtubules from one pole in prophase 1. Both chromatids of chromosomes are attached to kinetochrome microtubules from one pole in prophase 1. That is wrong, neither. Prophase 1, they don't attach at prophase 1. Sister chromosomes of chromosomes move to the same pole in meiosis of anaphase. Sister chromatids of one chromosome move to the same pole when any opposite pole. Genetically non-identical 4-daughter nuclei form. That is correct. Fifth one is the answer. Fifth is the answer. Question number 2. Kinetochore of each homologous attached to microtubules from one pole or the other. Kinetochores of each homologous chromosome attached to microtubules from one pole or the other. Kinetochore of each homologous attached to microtubules from one pole or the other. Prophase, metaphase 1, prophase 1, anaphase 2, metaphase 2. Kinetochore of each homolog attached to microtubules from one pole or the other. One pole or the other. Both kinetochores are attaching to same site. It has to be prophase 1. Then it has to be prophase 1, right? Attached to one pole or the other pole. Then prophase 1. Then people centromeres reach to opposite poles. Centrosomes reach to opposite poles. Centrosomes are reaching to opposite poles during the metaphase. Which of the above events can be taken place in the same phase of the eukaryotic cell cycle? Same phase of the eukaryotic cell cycle. Centrosomes reach to opposite poles. Kinetochore protein attaches to sister chromatids. Movement of chromosome back and forth. Back and forth take place in prometaphase. Kinetochore attachment also occur in the prometaphase. Chromosome become less condensed to form chromatin anaphase telophase. Centrosomes reach to opposite poles metaphase. So BC must be the answer. BC is the answer. Next one, in cells cycle, the centrosomes reach to the opposite pole during. In cell cycle, centrosomes reach to opposite poles during metaphase. They completely, the movement of the centrosomes are going to complete people at the metaphase. That is what they have mentioned. All anthophyta possess embryos with two cotyledons. Neither. Monocots are not having two cotyledons. Contain male gametes and pollen grain. All anthophyta contain male gametes and pollen grain. Male gametes in pollen grain. Correct. Pollen grain contain 3 apertures. Wrong. Because that's present in dicots. Monocots have 1. Have taproot system. No, that's in dicots. Fruits present. All anthophyta. Fruits present. Yes. මෙතන ළමයි contain male gametes the male nuclei ද male nuclei නේද? I think then the best answer goes for the fifth one, right? Why not male gametes? We said that male nuclei කියලා කිව්වේ. Male nuclei, right? They don't form cells there. Only two nuclei are going to form. Ne? So fifth one is better than people. Question number 6, correct statement about the plant meristematic cells and tissues. Correct statement regarding plant meristematic cells and tissues. There are three types of meristematic tissues in all plants. There are three types of meristematic tissues in all plants, Lou. There are three types of meristematic tissues: apical meristem, lateral meristem, intercalary meristem. Harine. Three types of meristematic tissues in all plants. හැබැයි all plants කියපුවම පොඩි අවුලක් තියෙනවා. බලමු අනිත් ටික නේද ළමයි? Dense cytoplasm and the central vacuole are characteristic. Correct එක අහන්නේ. Dense cytoplasm and central vacuoles are characteristic feature of all these cells. නෑ නේද? Central vacuole is not present. Wrong. All gymnosperm plants have lateral meristems. All gymnosperm plants have lateral meristems. එක හරි. All gymnosperms have lateral meristem. All gymnosperms are showing secondary growth. Meristems can divide under any environmental condition. There are certain conditions that makes the meristems dormant. After cell division, cell differentiation and cell elongation occur respectively. Cell differentiation comes third, the wrong order. Then all three types of meristematic tissues are present in all plants. Because intercalary meristems are in monocots, not in all plants. So I'll take people third one as the answer for the sixth one then. Third one for the sixth. Right, people, then we are moving to next question. Question number 7. Which of the following statement regarding the vascular tissue or of the plant is correct? Which of the following statement regarding vascular tissue plants correct? Phloem of Gnetophyta has sieve tube elements. Sieve tube elements, correct. Phloem of Gnetophyta has sieve tube elements. Gnetophyta has xylem vessel elements, not sieve tube elements. Wrong statement. Sieve cells of the Bryophyta transport organic food. Sieve cells of Bryophyta. Bryophyta don't have vascular tissues. Wrong. Sieve tube element have pits between themselves. Sieve tube elements have pits between themselves. Pits name plasmodesmata. Wrong. Seedless vascular plants do not possess sieve tube elements. Seedless vascular plants do not have possess sieve elements. Seedless vascular plants do not possess sieve elements. Seedless vascular plants do not possess sieve elements. Wrong. They have neither. Seedless vascular plants have sieve cells. Correct. Possess sieve elements. Sieve tube element කිව්වනම් වැරදි. Do not possess sieve elements. Anyway, මේ statement එක වැරදිනේ. Companion cells are found in Psychodophyta. Companion cells are found in Psychodophyta. Wrong, ne? No answer. Phylum of Gnetophyta has C2 elements කියන එක ගන්නත් බෑනේ ළමයි. ඒතර මම answer එක දීලා තියෙන first කියලා අපි all කියලා හදාගමු. හරිද? All. No answer for this one. Market scheme එක මම first කියලා අරන් තියෙන්නේ, but actually they have xylem vessel elements, not C2 elements. So that's a wrong statement. The phylum of non-vascular plants that has more recent common ancestors with seedless vascular plants. ළමයි ප්‍රශ්න තියෙනවා නම් අහන්න සද්ද නැතුව නිකන් ඉන්නපා. The phylum of non-vascular plant has more recent common ancestor with phylum of non-vascular plants. Vascular plants, non-vascular plants are having more recent. Non-vascular plants කියපුවාම we have people Bryophyta, Anthocerophyta, Hepatophyta, Anthocerophyta, right people? Anthocerophyta is having relationship. So fourth one is the answer. Then select the characteristics common to monocotyledonate and dicotyledonate from the below features. Characteristic to common to monocotyledonate and dicotyledonate. Having 3 types of dead cells in the xylem. Monocotyledonate and dicotyledonate having 3 types of dead cells. Tracheids, vessel elements, sclerenchyma. Hurry. Spores are developed into bisexual gametophyte. Spores are developed into bisexual gametophytes. Ne, can't be bisexual, ne? Unisexual. Megaspores of the megaspore mother cells undergo meiosis within the ovule. Megaspore mother cells undergo meiosis. Megaspore mother cells undergo meiosis within ovule. Megaspore mother cells undergo meiosis within ovule. Undergoes meiosis within ovule. Megaspore mother cells undergo meiosis within the ovule. Megaspore mother cells undergo meiosis within the ovule. Correct, Nedalame. Mother cells, they are present inside the ovule and then they undergo meiosis. 4 megaspores. 4 megaspores undergo then further division. Ne? 4 megaspores, 3 are going to die, 1 remain, it divide further. Coelid sperms are present. Coelid, neither, wrong. So I'll take A and C. Select the common kilahanne. A and C kilaganno. Second, ninth ke. Ninth ke second. 10th ke lamai question number 10. Which of the following correct statement about the plant tissue? Correct statement regarding plant tissue. All anthophyta have reticulate. All anthophyta have reticulate venation. The parallel venation present in some. There are more chloroplast in palisade cells in monocot leaves. More chloroplast in the palisade of the monocot. Vendabani monocots don't have palisade. There are cellulose microfibrils around the guard cells arranged vertically. Cellulose microfibrils around the guard cells arranged vertically. The radial is the radial. Ilagata Casparian strip is present on the vertical and the radial walls. Vertical and the radial. Transverse and the radial walls. Wrong. Guard cells are only type of epidermal cell with chloroplast. That is correct. Fifth one is the answer. Then people, question number 11. Question number 11. Xylem tissue consists of tracheids, fibers and parenchyma. Xylem tissue consists of tracheids, fibers and parenchyma. Correct. Production only one type of spores. Produce only one type of spores. Produce pollen grain. Possess erect stems. Common characteristic shown by Neplephis and Lycopodium from the above. Xylem tissue consider tracheid fibers and parenchyma. Correct. Produce only one type of spores. Neplephis, Lycopodium. Correct. Producing production of pollen grain. Wrong. They don't produce pollen. Possess erect stems which produce strobilus. Erect stem strobilus. No. Neplephis, you can't take strobilus. Wrong. So we'll take people A, B, A, B, A, B, A, B. What did they ask? Common characteristic shown by both Neplephis and Lycopodium. The xylem tissue consists of tracheids, fibers, and parenchyma, producing only one type of spores. Producing only one type of spores. What is your answer, people? We'll go for A and B as the answer. First one is the answer. Question number 12, people. Which of the following correct regarding plant tissues? 12. Which of the following correct regarding plant tissues? Parenchyma cells possess unevenly thickened secondary cell walls. No secondary cell walls. Wrong. All meristematic cells constantly divide and elongate and differentiate later. All meristematic cells constantly divide and elongate and differentiate later. All meristematic cells constantly divide and elongate and differentiate. All meristematic cells constantly divide, elongate and differentiate. ඒක හරි නේද? All meristematic cells නෑ නේද? Not all නේ. Some meristematic cells continue their division, some are transforming into some differentiate නේ. So all කියන එක වැරදි. This all is wrong. Guard cells of all plants leaves are bean shaped. Wrong නේ. Monocots are not bean shaped. And then monocots are having dumbbell shaped ones too. So therefore all guard cells of all plants can bear. Chemicals secreted by some trichomes involving in defense against herbivores. Ekari. Vessel elements of xylem tissue are wider and have thicker walls than the tracheids. Vessel elements of xylem tissue are wider and have thicker thinner walls. Wrong lama. Fourth one is the answer. Question number 12, fourth. Question number 13, people, parenchyma cells contain secondary walls deposited to give deposits. Parenchyma contains secondary wall deposit to give mechanical support without restraining the growth. Eka hari. Provide support in place where the growth has stopped. Wrong. Neither. Are generally elongated. Correct. Mithana secondary wall kine ke varadi lamai. Parenchyma don't have secondary cell walls. Only have primary. Ne? Composed of pith of the root of monocots. Composed pith, wrong. Ne? Parenchyma not present in the pith. Have irregularly thickened cell walls. Irregularly thickened cell walls. Irregularly thickened cell walls. So what do you say? Parenchyma corners are thickened. Irregularly thickened cell walls කියන එක කාටද අපි දාන්නේ? ඒක නම් පොඩ්ඩක් බලන්න වෙයි වගේ ළමයි. Irregularly thickened cell walls. Where did we discuss that? Irregularly thickened cell walls. Where was it? They say people parenchyma unevenly thickened. Irregular word එක පාවිච්චි කරලා නැහැ හැබැයි. Irregularly thickened කියලා නැහැ. Unevenly thickened කියලා තියෙන්නේ. Sclerites වලට බලමුද? එතන පොඩි doubt එකක් තියෙනවා ළමයා. මේ regularly කියන එකට unevenly, I think anyway the word is wrong. We'll go with the third one as the best answer for that. Ilana, question number 14. Question number 14. Correct statement regarding vascular tissues. Correct statement regarding vascular tissues. Vessel elements found only in angiosperms. Vessel elements found only in angiosperms. Vessel elements found only in angiosperms. Nane, the netophyta has two. Then water move freely through the perforated plates found in the tracheids. Water move freely through the perforated plates found in tracheids. Water move freely through the perforated plates found in the tracheids. Freely through the perforated plates found in the tracheids. Water move freely through the perforated plates found in tracheids. Wrong. Ne? Perforated plates are in vessel elements. Companion cells are found in seed-bearing plants. No. Some seed-bearing plants. Who has that companion? Flowering plants. Ne? Long, narrow sieve cells are found in the ferns. Long, narrow sieve cells are found in ferns. Hurry. Sieve plates are found in terminal of the sieve tubes. Sieve plates present in the terminals. Sieve plates are found in the terminals of the sieve tubes. Then sieve tube ke lage yani interconnected long tubes. Sieve tube element ke end only mitande. To wrong any sa. So we got the answer now, people. Fourth one is the answer. Question number 15, people. Collenchyma cells vary from parenchyma cells because containing thick primary cell walls. Because of containing thick primary cell walls. Because of containing thick primary walls. Collenchyma cells vary from parenchyma cells because of containing thick primary walls. Vary from parenchyma because of containing thick primary walls. Nani? Because of not containing secondary walls. Nah. Because of photosynthesis. No. Because of dead cells. No. Because of containing large central vacuole. Correct. Fifth one is the answer. Fifth one is the answer. So that's the end of this discussion, people. That's the end of the discussion. So make sure that you be ready with the sections. I'll be giving you paper on Saturday. So you bring the paper on Saturday class, okay? You bring this paper on the Saturday class and get paper 9, right? We will just control the waste of materials unnecessarily for the people who are not participating for the discussions and who are not writing and submitting. I think we should not waste our materials on them. So you make sure that you bring the tute. So I'll exchange. I'll give you the new tute for this paper number 8, right? So we'll exchange that. So that's good, right, for you. It's a what do you call privilege for you. And then next thing, people, you are definitely, you have to be happy about yourself because you are going to be ahead, definitely, because you are the people who are now managing the work. Most of the others don't manage because they are now they're not balancing the work. So that's the nature of this exam. Whoever people struggle hard, they can maintain it. Otherwise, can't maintain. So keep that in mind. Work continuously, people, non-stop manner, right? You will. It's a long journey anyway, right? You can't earn people good things in life that easily. You have to work hard and then work continuously to earn it for you. This will open the gate for you to a new world. So work for that, and I'll see you all people on Saturday class. Thank you very much. Right, a big salute for all of you for joining with the class today. See you. Bye.